Okay, so we're going to start with this next video. This is video number 2, and we're going to start with the cell cycle. This image shows you the complete cell cycle. It shows as a cell enters right here. This is the beginning of the cell cycle, and the cell can do one or two things. It can either exit the cell cycle and enter into cell cycle arrest, which is the G0 phase, and some cells, once they enter into this. For example, your nerve cells, if you damage your nerve cells, they don't replace or repair themselves because they've entered into the GO or the G0 phase. But most cells start into the cell cycle right here, and this represents all of this section interphase. So collectively, you can see right here it's labeled interphase. From here all the way to here is interphase, and it consists of 3 phases: G1, S, and G2. So in G1, basically the cell grows and it begins to increase the number of organelles that it has, and the cellular contents are just duplicated, but not the chromosomes. The chromosomes aren't duplicated until the S phase, and then replication occurs and a copy is made of each chromosome during the S phase. It then enters into G2, which this is just the second growth phase, and anything that needs to be repaired or any additional growth that needs to occur happens there. So you can see right here it starts in with the 2 single strands of chromosomes, so it has 2 chromatids. It enters into the S phase, and here it makes the X kind of structure where it's made a copy of itself, and it has sister chromatids now. It still only has 2 chromosomes, but it has a copied set of chromosomes. Then it enters into the G2, grows, gets a little larger, and that's where it enters into mitosis. So mitosis and cytokinesis collectively are called the mitotic phase or the M phase, and basically this is prophase, anaphase, telophase, and I don't know if you guys remember from last night, they had prophase divided up into 3 parts, so this is probably early prophase, prophase, late prophase, anaphase, and then telophase. So the nucleus is actually divided during mitosis. That's the main function of mitosis, is to divide the nucleus and sort the chromosomes out into 2 new cells at opposite ends of the cell. And then in cytokinesis, that's just the division of the cytoplasm. So here's where the chromosomes condense, line up in the center, move away from each other, and then begin to have a nuclear envelope form around them, so that it actually has 2 tiny nucleuses with chromosomes that you can see at the end in telophase, and then it's pinched into, and or else a cell plate's formed if it's a plant cell, and it creates a cleavage furrow if it's an animal cell, and the cytoplasm is divided in cytokinesis, and that creates 2 new cells. So you can see this is only mitosis, because mitosis only makes 2 daughter cells with the complete set of chromosomes. It went from a structure that had a duplicated set to where they each have 1 of the sister chromatids. And here are the phases of mitosis. I put the little star in there because they had more than 1 description for prophase, but for your purposes you just need to know prophase, metaphase, anaphase, and telophase in that order. We also looked at onion root tip cells last night, and we found a lot of cells that had no chromosomes visible. They just looked like regular plant cells like we had seen in our previous labs, and that's because 90% of the cell life is in interphase, which is G1, S, and G2. So you cannot see the chromosomes at that point. You can see the nucleus pretty clearly, but no individual chromosomes, and that's interphase. And then each of these phases have particular chromosome structure, and that'll be in the next few slides. Sorry about that, that was our bell. Okay, so here are all the phases, and you can see they've put in one of the phases of interphase, and you can see the chromatin is duplicated inside of the nucleus, and you can't see the individual chromosomes because they haven't condensed yet. So this is interphase, and the cell is just growing and preparing to divide. Some of you guys on your handout for the lab said nothing was happening in interphase, but that's not true. You need to think everything that it does to prepare to divide occurs in interphase. So growth and duplication of chromosomes are the main things that occur in interphase. So in G2 of interphase, it looks like this. There are the centrosomes, each with a pair of centrioles. This is something that has to duplicate too because they're going to migrate to opposite ends here in prophase. You can see that the mitotic spindle, or the spindle fibers, are coming off of the centrioles, and the nuclear envelope is beginning to disintegrate at this point, and the chromosomes are condensing and becoming visible. This is one of the stages that you're not going to be required to know, but you need to know that it's part of prophase. Either one of these are considered prophase because here you can see the nuclear envelope has broken up more. The chromosomes are more condensed and more easily seen, and you can see where the spindle fibers are actually attaching to the centromeres of the sister chromatids, preparing to line them up in the center. So prophase. Then we go to metaphase. Remember, meta means middle, and so you can see the chromosomes are lined up just like in a football game, right at the on the line, perfectly where their sister chromatids are able to have one of them go this way and one of them to go this way. And this is metaphase, in the middle. No nuclear envelope exists. It's totally been disintegrated, and its parts are still there. It'll later reform, but it's not visible. Next phase is anaphase, and remember, ana, away, and that's when the sister chromatids move away from each other. And as soon as they move away from each other, they're now called daughter chromosomes, and they are a chromosome in their own right. Telophase, the nuclear envelope begins to reform. The cleavage furrow in animal cells begins to reform, and it's going to look like two new cells completely. You can still see the chromosomes, and it hasn't completely pinched in two because that's in cytokinesis, where the cell splits into two new cells. This is what our root looked like last night under the microscope, and you could see all of the tiny nucleuses. Sorry, there's our bell again. And this was under further magnification, and you can see right here the chromosomes are very visible, and they're lined up in the middle. They're lined up in the middle here, and right here in this one you can see they're beginning to pull away from each other. So let's see, you can see there's no chromosomes clear in like either of these two cells, so this would be an interphase. And whenever you begin to clearly see the chromosomes again, see, you can see chromosomes here and here. You can even start to see chromosomes here. Then those are prophase cells. Anywhere you can see chromosomes, then that's a prophase. Then they line up in the middle, which is metaphase, move away from each other, anaphase, and then in telophase you kind of have to be careful because you'll still be able to see chromosomes, and the cell will actually take up as much space as one of the cells that was in interphase, but it's going to have two tiny nucleuses. So here's one, here's one, this one could be one here. It's kind of hard to tell for sure, but some of them are really clear. I would say there's no doubt with this one right here because it's a tiny cell and you can see two nucleuses clearly. So one of the things that you're going to be asked to do is to take a microscope, and I'm going to hand you an onion tip slide. You're going to have to get to the 40x objective, and you're going to have to find at least two phases and identify them, and I'm going to have to come over, look, you're going to have your little pointer pointing on it and say this one is metaphase, anaphase, telophase, interphase, etc. We also looked at fish blastulas, which are just basically little eggs, and so you can see here are the eggs. We saw this on the red objective and the lowest objective, and you could actually see, oh, okay, that could be just like a ball of cells. And then as you zoomed in and you got down to the highest objective, you could actually see the chromosomes. Our cells were not this clear. I think I saw one person who had cells this clear, but you can tell very distinctly, and they've got them numbered for you. 1 is prophase, 2 metaphase, because they're in the middle, and you can see the spindle fibers. You can see the aster that's forming here. 3 anaphase, and here it's beginning to cleavage furrow, so this would be 4 telophase. So I'm going to go through each one of these, and I'm going to see if I can give you a description along with what it looks like in the slide. Okay, so here's the G2 of interphase. Just like we saw before, the chromosomes are not condensed. You can't see them, and it would look kind of like this. You can still see the nucleolus. You can clearly see the cell wall, and you can see the nuclear envelope still intact. Prophase. This is something that you might note right here. This would be the description that you give for prophase. So chromosomes become visible, nuclear envelope dissolves, and the spindle forms. And the chromosomes consist of 2 sister chromatids because they're duplicated chromosomes. So you can see right here, prophase that's early and prophase that's late. We're going to look at this one, even though you don't have to understand the difference between prophase and prometaphase, but you might see chromosomes like this that's late into prophase, and basically it's just that they have spread out and the nuclear envelope is not visible at all. Metaphase, they line up in the center, and this isn't even as clear as some of those we saw in the slide before, but that's the way you know is there's spindles attached to chromosomes that are lined up in the center. That's metaphase. In anaphase, the centromeres, which is the central structure of 2 sister chromatids, divide, and the chromatids are now called chromosomes and move toward opposite poles. OA, anaphase. Here is an example. Notice you can see 2 tiny nucleuses pretty close together, taking up almost as much room as one of the cells that's in interphase. And you can even see the cell plate or the beginnings of a cell wall form. So chromosomes uncoil, nuclear envelopes reform, and cytokinesis divides the cell right directly after telophase. So this is the last note I'm going to give you with this one. I'm going to end this video, and then I'm going to go to the slide that goes with your books. So a comparison between mitosis and meiosis. Everything we've talked about so far has been about mitosis. The only thing I mentioned about meiosis is that it occurs in germ cells and produces gametes, and there's a whole slideshow just for meiosis, and I'm going to wait till we're in class to do that one. I'm just going to cover mitosis today. So mitosis occurs in somatic cells, and it's to replace and repair cells. It only produces 2 identical daughter cells, and it only has 1 nuclear division. And the daughter cells are diploid, which means they have a full set of chromosomes. So if you were in lab last night, you got to draw out the stages of meiosis, and this might make a little bit more sense. If not, you have your textbook, and half of the chapter 5 is on meiosis, and you can look at some of the images and kind of get an idea. This occurs in germ cells and produces gametes, which another word for gametes are just the egg and the sperm. Those are your gametes. It produces 4 daughter cells instead of 2. It has 2 nuclear divisions. It has prophase 1 and prophase 2, metaphase 1 and metaphase 2, and so forth. And daughter cells are haploid. They reduce the chromosome number to half. So that is the end of that, and I'm going to go ahead.